guys, and welcome back to Biologue. Hope you're having a great day and a great week. Today's video is going to be about plant and human nutrition. So we're going to look at plant nutrition and human nutrition topic wise questions. If you like this video, do like, share and subscribe to Biologue. With that being said, let's just get right on to these questions. Okay, state the balanced chemical equation for photosynthesis. This is self-explanatory. You have C6H12O6 as glucose. This is glucose, so 6CO2 plus 6H2O under the presence of light and chlorophyll give you C6H12O6 plus 6O2. Make sure you write this part because this carries 1 mark. Alright. A student investigated the effect of different wavelengths of light on the rate of photosynthesis of water plant Camboba. The student used the apparatus shown in the figure. This is the figure. Now we have 1% of sodium hydrogen carbonate solution which releases carbon dioxide. So remember two things, okay? The first thing is soda lime. Soda lime is something that absorbs carbon dioxide. So it's usually used to suck in or absorb the carbon dioxide. Second is this, which is the sodium hydrogen carbonate. Okay, NaHCO3, sodium hydrogen carbonate, which will give you produce the carbon dioxide. It will release or produce carbon dioxide. Alright, so the student collected the gas produced by the plant for 5 minutes and the results are shown in table 6.1. Describe the effect of wavelength on the rate of photosynthesis as shown in the student's result as shown in table 6.1. You will gain credit if you use data from the table. So this is actually a very good indication that you have to use the data given in the table, otherwise you won't get good marks for this question. So you will gain credit if you use data from the table. You have to use data from the table. That is what it means. So let us look at the table right now. So we have violet as 400 wavelength 0.8, blue 475 0.8, green 550.2, yellow 600.4 and red 675.9. Okay, so the effect of wavelength on the rate of photosynthesis is as the wavelength increases, we go down, we see wavelength is increasing, the rate of photosynthesis initially it will decrease. So if you see here till about this part, till about 550, the rate of photosynthesis is actually decreasing because volume of gas collected is decreasing. But beyond this point, it starts to increase. So this point, the rate of photosynthesis is increasing. As wavelength increases, the rate of photosynthesis decreases till 550, but after that it will increase. So it increases beyond this wavelength, right? Second one, maximum rate of photosynthesis is at 675. That is true. We are able to see that the maximum rate is at 675. 0.9 is the volume of gas collected in centimeter cube. So the maximum rate of photosynthesis is at 675 Newton meter wavelength, at 0.9 centimeter cube volume of gas collected. Highest rate is in violet, blue and red. Okay, highest rate is in violet, blue and red. Let's see. So when we say rate, we have to look at the volume of gas collected. That will show us the rate of photosynthesis. So violet has 0.8, blue has 0.8 and red has 0.9. Compared to the other three, you're able to see that violet, blue and red have a greater volume of gas collected, hence have a greater rate of photosynthesis, right? So it has a higher volume of gas collected, so greater rate of photosynthesis. State how the student would calculate the rates of photosynthesis from the results in table 6.1. Right, the rate, it's called rate of photosynthesis, okay, which means the amount of gas collected, the volume of gas collected divided by the time taken to collect that gas. So rate of photosynthesis is the volume of gas collected divided by the time taken. Okay. Let us see the next question. State why the student kept the lamp at the same distance during the investigation. This is to allow for the same light intensity and to ensure that light intensity is a controlled variable, right? Because if it is not a controlled variable, it would affect the results. So you will be able to see that not only the wavelength is affecting it, but the light intensity is also affecting it. So you need to ensure that the light intensity is the same by keeping it a controlled variable, so that only wavelength is affecting the result. Only wavelength will be affecting the rate of photosynthesis. Used sodium hydrogen carbonate solution, right? We mentioned sodium hydrogen carbonate solution is used to produce or to release carbon dioxide, so that only the wavelength of the light is the limiting factor, right? So only the wavelength of light will be the factor that limits or affects the rate of photosynthesis. State 3, uses in a plant of the carbohydrate produced during photosynthesis. So it is used to produce cellulose in the cell walls, used in respiration to release energy, to produce starch for storage. You can also mention other things, for example, sucrose for transportation, and you can also talk about the lipids. Sometimes it's used for lipids, it's used for amino acids and things of that nature, but these would be the most precise answers. Okay. The next question is, a student carried out an investigation to find out the effect of carbon dioxide concentration on the rate of photosynthesis of an aquatic plant. The apparatus that the student used is shown in the figure. The student was advised to use a light meter positioned at the same distance from the light as the pond plant. The student counted the number of bubbles produced by the cut end of the stem. Okay, so that is the diagram. Explain why the student included the following apparatus: the beaker of water and the thermometer. So the beaker of water we see is actually used as a heat filter. So it is used to maintain a constant temperature. Okay, it's used to maintain a constant optimum temperature, and it acts like a heat filter, right? It acts like a heat filter because temperature here is a control variable. You need to control it, right? You do not have to change the temperature, because here only the carbon dioxide concentration is being changed. This is the dependent variable. So always when you're doing these kinds of questions, first mark the dependent and the independent variable. So since here only carbon dioxide concentration is what is affecting the experiment, affecting the rate of photosynthesis, the temperature will have to be a controlled variable. So thermometer will be used to measure the temperature of the water. Light meter and the ruler, why is it used? Light meter is used to maintain a constant light intensity. This word intensity is important. So it's used to maintain a constant light intensity, and the ruler is used to ensure that the lamp is always at the same distance from the light source. Okay, that the lamp is always at the same distance from the plant and from the light source, so that the lamp is always at the same distance from the plant, right? Sorry, not the light source. I mean the lamp will always have to be at the same distance from the plant, so that light intensity is the same. So the light intensity will be the only dependent variable. All right. So here it says that light intensity on the plant is dependent on the distance from the lamp, right? So as you move the lamp further and further away from the plant as such, so let's say you have your plant and this is your light source. All right, so let's say you move the lamp further and further away from the plant, the intensity decreases, and if the intensity decreases, the rate of photosynthesis decreases, because light is needed for photosynthesis. Alright, the next question. The result obtained by the student is shown in this figure. Now you have to describe the student's results, right? This term, describe, you have to know the difference between describe and explain. Describe means to just pick up the information from the graph and state that in the form of an answer, right? Just pick up points from the graph. Explain means describe it, but in terms of biological concepts. So you have to show that you have knowledge of the terminology, the biological concept as such is what is meant by explain. So when they say explain this, you have to mention in terms of biology as such. Okay, so you will gain credit if you use the data in the answer from the figure. Okay, rate of photosynthesis increases as carbon dioxide concentration increases, right? In this part specifically, we can see rate of photosynthesis is increasing with the carbon dioxide concentration. Alright, so if we look at this, the next point is that it levels off as the graph plateaus. So when we say graph plateaus, we mean this region. Alright, This region is where the graph plateaus. So this is where the graph levels off. Alright, and the concentration, which is the rate of photosynthesis, actually the rate of photosynthesis, it remains constant. Okay, so rate of photosynthesis increases as CO2 concentration increases, and that is till this point. But after that, the concentration actually remains the so it keeps on increasing. So the concentration keeps increasing, but the rate of photosynthesis remains constant. So the graph plateaus or levels off. Rate increases to 200 bubbles per minute or 0.4, and this is basically the final thing. Okay, so we are talking about the overall change. We are talking about overall change. What is the overall change in the rate? So it increases to 20 bubbles. Okay, and this is the maximum that it reaches, the peak. Till 0.1 concentration, rate of photosynthesis increases slowly. We are talking about this portion of the graph where till this part only a slow increase is there, but here we are able to see a steep increase, and then here the graph plateaus or remains constant, or we can say it levels off. State the limiting factor that is limiting the rate of photosynthesis in the region X of the graph. Region X is where there's a steep gradient, which means that this factor in the bottom, this independent variable, is still affecting the rate of photosynthesis. Only in this portion where the rate of photosynthesis no longer changes, even if there is a change in the carbon dioxide concentration, only in this region the carbon dioxide concentration is no longer a limiting factor. So only in that region there is another factor, some other environmental factor, that is the limiting factor. Alright, state and explain the reason for the shape of the graph in region Y. In region Y is the plateau where the graph remains constant. So carbon dioxide concentration is no longer the limiting factor, and it is another environmental factor like temperature could be the limiting factor. This could be because temperature influences the rate of enzyme activity, right? And enzyme activity will affect the rate of photosynthesis. So rate of photosynthesis depends on it. Since the graph plateaus, the light intensity could also be a limiting factor, and this will also affect the rate of photosynthesis, right? Because light is needed for photosynthesis to occur. The next question, counting bubbles may not always be the best way to measure the rate of photosynthesis, because the volume of the bubbles may not always be the same. That is true. So the volume of bubbles may vary. Alright, so suggest and explain one alternative way of measuring the gas given off to solve this problem. So to measure the volume of the gas produced, we can either use a gas syringe or an inverted measuring cylinder filled with water. So gas syringe is an instrument that actually looks somewhat like this. It looks like a common syringe, but it has the graduations, and you'll be able to measure the volume of gas using this. You'll be able to measure the volume of gas. So you can use an inverted measuring cylinder also, which will be filled with water. So then the amount of volume of the water displaced will be equals to the volume of the gas collected. Here the gas collects at the top and causes displacement of the water. So the displacement of the water, how much volume of water that is displaced, is equals to the volume of gas collected. The gas syringe needs to be attached to a delivery tube, right? And this is what we call a delivery tube. Alright, you'll have something like this. This is a cork, and we have the thing. So this is the delivery tube. So let's label this. This is the delivery tube. That is the gas syringe. This is the cork. We call it a cork. And this is the conical flask. So this is how your experiment will look. Alright. Now figure 2.3 shows the carbon dioxide concentration in the atmosphere as determined at Mauna Loa in Hawaii between 1959 and 2013. So this is the graph. Explain why the concentration of carbon dioxide has increased between 1959 and 2013. Alright, so why does concentration of carbon dioxide increase? This is because of greater combustion of fossil fuels, greater burning of fossil fuels, increased use of appliances like cars and air conditioning, right? Because cars, the exhaust, the engine causes carbon dioxide to be released to the atmosphere, and also release of carbon dioxide from volcanic eruptions. Alright. Now moving on to the last question. Global warming is largely due to an increase in carbon dioxide concentration in the atmosphere. Atmospheric carbon dioxide concentration. Explain how increases in the atmospheric carbon dioxide concentration contribute to global warming. Alright. So since carbon dioxide concentration is, or carbon dioxide itself is a greenhouse gas, due to the greenhouse effect, there will be an increase in the trapping of heat or infrared radiation. So these rays will be trapped and absorbed by greenhouse gases, and when they are emitted as long wavelength radiation, which cannot escape from the atmosphere, right? So the incoming rays are in fact short wavelength rays. So like in the case of a greenhouse, when you see in a greenhouse, why is it that in a greenhouse the temperature is able to remain high? Because it traps heat. It traps a short wavelength infrared radiation that is coming in, and it emits it as long wavelength infrared radiation. So the long wavelength rays cannot pass through the glass, right? The long wavelength rays will be difficult to pass through, so they remain in the atmosphere. That is exactly the type of phenomenon that they are using here, which you have to describe in detail. And in terms of carbon dioxide concentration being trapped in, or carbon dioxide as a greenhouse gas trapping the heat and then releasing long wavelength infrared radiation, which gets trapped in the environment, and so it cannot escape from the atmosphere. Alright, let's move on with the first question. Australia has added fluoride to much of its drinking water since 1953. Other countries, such as Chile, do not add fluoride to their drinking water. Outline the arguments for and against the addition of fluoride to public drinking water. So firstly, fluoride is cheap to supply and it is widely available for use. That is the first point, right? So it is cheap to supply and it's also widely available, so it's easy to get is what the main point here is, right? The second point is it helps strengthen the tooth enamel. Yes, that is true. It helps strengthen the tooth enamel. So the first point actually is that it helps strengthen the tooth enamel, and because it is cheap to supply as well as widely available for people to use, it is seen as one of the common ways to actually make the drinking water more suitable for people, or add fluoride to drinking water itself, right? Third point is that it may cause allergic reactions. This is the arguments against the use of fluoride. Why shouldn't you use fluoride? You shouldn't use fluoride because it may cause allergic reactions or allergies in some people, as well as it may cause certain problems for certain people if excessive use is there. Obviously, if anything is used in excess amounts, it will cause some problems. So something you can write over here is that it may cause the discoloration of teeth, or fluorosis. It's called fluorosis, which is basically almost a yellowing or discoloration of teeth. Now, studies of the relationship between sugar, tooth decay, and fluoridation of drinking water have been carried out. Data was collected on the tooth decay in 12-year-old children in Australia and Chile. Now the figure shows the changes in the sugar consumption in Australia and Chile between 1970 and 2006. Figure 5.2 shows the changes in tooth decay in the same countries over a similar period of time. Let's look at the diagram. So this is the diagram for sugar consumption, and this is the diagram for tooth decay. Alright, so this portion is Chile, this portion is Australia. So this is Chile and this is Australia. Okay, so the next question is, describe the changes in sugar consumption and tooth decay in Australia and Chile between 1970 and 2006. Now this is a graph question. It's a very important question. So you should actually look at the trends, right? It's not necessary for you to always point out the specific details in the graph, because most of the times what the examiner they expect from you is to understand the trends in the graph, the patterns, what exactly is happening on an overview. Okay, when you first glance at the graph, what do you see? So if we look at the sugar consumption graph, we're able to see that in Chile, alright, this is the Chile one. So in Chile you're able to see an increase till 1997, but after 1997 there is a decrease, right, till 2006. So that is your first point. Alright, in Chile it is increasing. It increased till 1997, but it decreased slightly after 1997, right, decreased slightly. Okay, Next point, in Australia it increased till 2000 but it decreased until 2006. So let us look. Let's say in Australia, right? So in Australia it increased. Yes, it increased till 2000 but it slightly decreased till 2006. That is correct. Now in child it decreases. This is, now we are talking about to decay, the second graph. In child it decreases from 1997 to 1990, but from 1990 to 1995 it increases. Okay, so now we are looking at this graph. It usually, initially it will decrease. Okay, this is for child. It decreases till 1995, but from 1990 to 1995, from 1990 to 1995 you are able to see increase. Then again from 1995, another point, the third point you could have added here is that from 1995 it again decreases till 2006. The last point is for Australia. For Australia it keeps increasing, it keeps decreasing from 1977 onwards. So if we look at that, this is the curve for Australia. So it keeps decreasing. You're able to see that there is a decrease. So let us erase all of these lines right here. Look at the light blue line. So you're able to see that the white portions over here, they keep decreasing till 2006. So yes, that is correct. Moving on to the next question. The peaks for sugar consumption and tooth decay in 12-year-old children and child occurred at about the same time. It has been suggested that an increase in the sugar consumption in children has caused an increase in the tooth decay. Explain how an increase in sugar consumption may cause an increase in the tooth decay. Okay, so firstly you have to mention what exactly happens when you consume sugar. So when you consume sugar, the sugar will remain on the teeth. It will stick to the teeth. Sugar remains on the teeth. Bacteria feeds on the sugar that is present on the teeth, right? It will feed on the sugars. It will use the sugar for its respiration. So when it uses it, the bacteria will respire aerobically. Okay, and the fourth point is, when it respires aerobically, it produces an acid. That is the most important point. It produces an acid, and this acid dissolves the tooth enamel and dentine. So it dissolves the tooth enamel and causes it to become more weak, and it softens the dentine. The enamel is, remember, the hard outer layer. It's the first layer. The dentine is the second layer right below the enamel. Alright, figure 5.1 shows that the sugar consumption in these two countries are similar. Figure 5.2 shows that the changes in tooth decay in 12-year-old children. Okay, now suggest explanations for the similarities and differences in tooth decay in the 12-year-old children in Australia and child. So what is the similarity and differences between both the cases in Australia and in child? Firstly, so let's look at the Australia and child, right? Yeah, so tooth decay decreases in both countries as time progresses. Let's look at the graph for tooth decay. So we are able to see overall, if we look at the overall change, this is 1, this is 2. Both countries we are able to see a decrease in the cases of tooth decay. So the cases of tooth decay decreases as time progresses. This is because, so what is the reason behind this decrease in the tooth decay cases? This is because of better oral hygiene, better dental care. So better dental care or better oral hygiene can be one point. Consumption of less sugary and carbonated drinks for children, as well as use of fluoride toothpaste in appropriate amounts, appropriate quantities. Yes, that is correct. Then why are the differences happening? The differences over here is that tooth decay in Australia. Let's look at Australia. So tooth decay in Australia, all right, which is the white portion, right, it's lesser. Overall, the amount of tooth decay in Australia is lesser than the amount of tooth decay in child, right? We're able to see that from the graph. So differences is tooth decay in Australia is lower than in child because of better dental services. Right. You can also mention about education or awareness about dental care, etc. Those could also be good points to add. That's it for today's video, guys. I hope you liked the video. If you did, do like, share and subscribe to Biologue, and I will see you guys in my next video. Have a great day.